Hello everyone. This session is going to be on regulation in mitosis, especially beyond the metaphase anaphase checkpoint, which is the third checkpoint in the cell cycle. Now, what is important to understand as we move ahead with this session is that until the metaphase, it is the CDKs, that is the cycle-independent kinases that have had a role in regulation, while from the metaphase anaphase stage, it is the anaphase promoting complex that has a role to play in regulation. So most of the regulations are that happen are because of the decrease in the cyclin dependent kinases and the increased activation of the anaphase promoting complexes. So let us look at the learning outcomes of this session. As the MCDK, that is the mitosic stage, cyclin mitosic stage CDK, sets the cell at metaphase with the sister chromatids ready for separation. So the MCDKs kind of ensure that the sister chromatids are aligned at the equatorial plane at the metaphase stage. And it is at this point that the anaphase promoting complex, which is already present in large amounts but is present in the inactivated state, gets activated. And so as it gets switched on, the system then moves from metaphase to anaphase. The cohesins are a set of molecules that hold the sister chromatids together, and these cohesins prevent the pulling of the chromatids towards the two poles. So in the anaphase stage, that is what is happening, that the chromatids are pulled towards the two poles, and it is cohesin that holds it and doesn't allow it to move towards the two poles. So what is very important, therefore, as the cell moves from metaphase to anaphase, is that this cohesin has to be removed, and it is removed by the anaphase promoting complex cyclosome system to allow then the sister chromatids to be pulled apart. The segregated chromosomes are then packaged into daughter nuclei at the telophase, and the nuclear membrane components reassemble due to the M cyclin dependent kinases getting lesser and lesser in number. Dephosphorylation of the MCDK targets is a key step to completing the mitosis. So let us look at what is really happening from metaphase to the stage where the cell enters the interphase again. So this session will also include cytokinesis. Now let us look at the control at the metaphase anaphase checkpoint. Now, one of the important things that is there in the metaphase is that all the sister chromatids, condensed sister chromatids, are aligned at the equatorial plane, and it is held together because of the spindle fibers binding to the kinetochore. And as it moves from metaphase to anaphase, what you distinctively are able to see is that the sister chromatids are separated and they are pulled towards the two poles. So this is what is happening at the anaphase stage. So therefore, in order for this to happen, there are few things that have to be regulated. So the sister chromatids which are present in the anaphase, at the metaphase at the equatorial plane, are held together by a set of molecules that is called as cohesins. We all know that cohesins wrap around the sister chromatids right from the S phase. As the DNA duplicates and you have homologous pair of the chromosomes present, immediately the homologous pairs are held together by cohesins. So this is a multi-complex system which is able to wrap the two sister chromatids together, and the sister chromatids always stay together because of the cohesins. So because they are held together, it helps the chromatids to condense to form such condensed pair of chromosomes. And therefore what happens is, because they become condensed and remain as sister chromatids, when they are pulled apart, then there is no entanglement of the chromosomes happening, and the two sister chromatids are very separate in their identity, and therefore they can be pulled apart easily. So in order for the sister chromatids to be separated, they have to be, the cohesins have to be removed, and that is aided by the APCC complex. So what happens is that the APCC is able to remove the cohesins. So let us look at how this happens. Now, since APCC is the one that is responsible for removing the cohesins, the inactive APCC complex has to become active. And that happens because of a protein that is called as CDC20. Now, the CDC20, when it is phosphorylated, and it is in fact phosphorylated by M-cyclin dependent kinases, and these M-cyclin dependent kinases, or even SCDKs for that matter, so when the CDC20 becomes active, it binds to the inactive APCC, and immediately after it binds to the inactive APCC, it becomes an active APCC. So CDC20 is key to the activation of the APCC complex. Now what happens is that there is a protein that is called as separase. This protein is the one that is going to break the cohesin and remove the cohesin. Now, until the metaphase stage, even if there is separase is present, it is rendered inactive because there is another protein called securin that binds to this separase. As long as securin is bound to the separase is inactive. So a securin separase complex keeps the separase inactive. And if separase is inactive, cohesin is intact. If separase becomes active, then cohesin is removed. So by far what we understand therefore, that until the metaphase, when the M phase is active but the APCC is inactive, the securin is always bound to the separase, and hence you have the separase always inactive. And so therefore the two sister chromatids will be held together by cohesins, and when they are bound to by cohesins, the two sister chromatids cannot be separated, cannot be pulled apart. Now, when the APCC becomes active, we all know that APCC acts by ubiquitination. So it would ubiquitinate the securin, and the securin is now going to get degraded in a proteasome. So therefore, by a proteasome ubiquitination method, the securin is degraded, and with the securin getting degraded, it is no longer bound to the separase, and so therefore an inactive separase becomes active separase. Now this active separase can actually remove the cohesins. So what you find in the red is the cohesins. So the separase can break all these cohesins, and so the sister chromatids can get separated because now they are no longer held together by the cohesins, and these sister chromatids can now be pulled to the two poles by the mitotic spindle that is bound to these sister chromatids. So that is exactly how, when the cell is moving from metaphase to anaphase, the active APCC ensures that the sister chromatids are not held together by cohesins, and that it does by activating the enzyme separase. And how does it activate the separase? By degrading the securin. So you can see how the regulation is detailed, and it is taken care that until APCC is inactive, the securin is not going to allow the separase to act, but the moment the APCC becomes active, it is allowing the separase to become active. So therefore, at that point where regulation of APCC to get active is so important for the anaphase to happen. If the APCC does not get active or the securin does not get degraded, then you will not have the pulling of the sister chromatids to the two poles. So that is something that is important. And also what is important is when the active APCC becomes, the APCC becomes active, it also starts ubiquitinating MCDKs, and the levels of MCDK will go down. And if the levels of MCDKs goes down, then what is going to happen is that the targets that are phosphorylated will now be dephosphorylated by activated phosphatases, but there is no longer enough MCDK to activate the targets further. So therefore, one of the key regulation points is also the fact that several of the CDK targets are getting dephosphorylated by important phosphatases. So this is how this is one way of one regulation that is happening at the metaphase anaphase checkpoint. Now to move on further, if you have chromosomes that are not detached, that is, the cohesin is still wrapped around the sister chromatids, then the sister chromatids do not are not pulled towards the two poles. And also one other thing that is regulated, or what is important or regulated, is the fact that whether the chromosomes get more detached or less detached also depends on the tension or the force with which the sister chromatids are pulled towards the two poles. So sensing tensions is one of the ways of sending a feedback to APCC to ensure to ensure whether the two sister chromatids are pulled apart or not. So let us understand this. Uh, this is the sister chromatid, and at the centromeric region, the centromere is bound to by the kinetochore. So this entire complex is the kinetochore. So you have the inner kinetochore. To the inner kinetochore is bound certain proteins that are called as aurora B kinase, and these aurora B kinase also have a regulatory role. And the outer kinetochore typically has what you see in blue, the NDC80 complex. Interestingly, when the tension is low, that is, the pull of the spindle fibers on the sister chromatid is low, then what is observed is that not many mitotic spindles are bound to the NDC80. So in such a situation, therefore, the aurora B kinase gets access to the NDC18, and because they get access to the NDC80, they phosphorylate the NDC80. So if the NDC80 is phosphorylated, its attachment to the mitotic spindle is less. So in simple words, if aurora B kinase is able to phosphorylate the NDC80, then the NDC80 is not able to attach to the mitotic spindles. So more phosphorylated the NDC80 complex, less is the attachment of the sister chromatid to the mitotic spindle. So therefore, it is very clear that you will not have the sister chromatids going to be pulled towards the pole. But when you have a high tension, that is, the force is greater, then as you can see, the NDC80 complex, the outer kinetochore region, is more pulled towards the mitotic spindle, and so the aurora kinase does not find access to the NDC80, and hence you will have the NDC80 not phosphorylated. So when it is not phosphorylated, it binds or attaches to the mitotic spindle or the mitotic spindle fibers more strongly, and therefore the pole is going to be further enhanced. The pull is going to be further enhanced. So this is something that is also observed as a regulatory mechanism. So a kinetochore that is not attached to the spindle fibers sends a signal back to block CDC20 APCC activations. So in effect, what it mentions is that if there is a low tension, then APCC does not get active. You see, but if there is a high tension, then APCC gets active because a signal is sent for the APCC to get active. And the signals can be passed on by several proteins, like for example, MAD2. So there are a set of proteins that actually send a signal to the APCC and the CDC20 to activate the APCC. So, these signal molecules are based on whether the tension is high or whether the tension is low. So you can see how different players are important in ensuring that you have activation of the APCC, and when the activation of APCC happens, then you know that the sister chromatids will definitely be pulled apart because the cohesin will be removed. Between the two sister chromatids, the cohesin will be removed. Now you must understand that this is just one part of the sister chromatid. You will have another sister chromatid over here attached to a spindle fiber on this end, because you know that two sister chromatids are held together. But here, for convenience, just one sister chromatid has been shown in the figure. Now moving forward, once it has to go from the equatorial plane to the two poles, there are two ways by which you have the two sister chromatids pulled towards the poles. One is what happens in anaphase A. So the microtubules start depolymerizing, okay, except the interpolar, inter polymers, mitotic spindles. The other mitotic spindles or microtubules are beginning to get depolymerized, and as you know that the mitotic spindle is bound to the kinetochore, as this mitotic spindle shortens in length. So depolymerization basically means shortening of the spindle fiber. So therefore, you would have the sister chromatid that is bound to this mitotic spindle being pulled towards the two poles. So one, you know, polewards movement is because of microtubule depolymerization. So that is one way by which you have the two sister chromatids pulled towards the two poles. The other way that happens in the anaphase B is this, that the poles are pulled apart. You can see that the centrioles that are present on the two, they are stretching away from each other, and as they stretch away from each other, the two sister chromatids are going to get separated. So this is another way. So here the main role is being played by a molecular motor, which is the kinesin 5. So you can see how the poles are moving apart. In all of this, what is very important is that the CDK targets have to be dephosphorylated, and that is obviously happening because the APCC is becoming active. And since the APCC becomes active, all the CDKs are degraded, and since the CDKs are degraded, no more phosphorylation of its targets happen, and so the available phosphatases will start dephosphorylating the targets. Then we come to what happens at the telophase. So at the telophase, as you can already see, you have the sister chromatids getting into the nuclei. So you can see that the nuclear membrane is forming again, and you can see that a contractile ring starts to form, to contract, to form a furrow. So the beginning to cytokinesis is already seen in the telophase stage. So disassembly of the mitotic spindle. So you can see that slowly, gradually, you would have a lot of mitotic spindles getting degraded, but only a few interpolymer microtubules remain. Otherwise, most of the microtubules start to get disassembled. And as you can see, there is formation of the nuclear envelope. So the dispersed nuclear envelope membranes actually associate with the chromosomes, and the nuclear lamina is assembled with nuclear pore complexes 2 getting incorporated. So effectively what we understand, that the nuclear membrane that had got disassembled during the pre-metaphase stage is now getting assembled again at the telophase stage. And this primarily happens because the lamins that were phosphorylated by the MCDKs are now no longer phosphorylated because MCDKs have decreased, and phosphatases are able to dephosphorylate the lamins. So when the lamins get dephosphorylated, you have the nuclear membranes again getting assembled to form a complete nucleus. And within the nucleus, as you can see, you have the sister chromatids assembled. So this is something that is happening at the telophase stage. Now we move on to cytokinesis. In the cytokinesis, as you can see, the contracted ring that had just formed in the telophase now becomes more pronounced, and that is because the actin-myosin filaments are forming a contractile ring, and it forms a contractile ring around the interpolar microtubules. So the interpolar microtubules are just giving a definitive base for the cleavage furrow to be formed. You can see that because of the myosin, you will have the contractile ring contracting, and it becomes more contracted. Because it becomes more contracted, the furrow is getting more defined. And so you can see how the two membranes will now come together, and you will have the two cells getting separated. Now, all this happens because of a set of regulatory molecules, of which one of the most prominent molecules is the Rho-A. Now, the Rho-A is a G protein and belongs to the family of RAS family. The RAS is again a family of G proteins. They are single proteins, single subunit G proteins. Now, what do we mean by G proteins? G proteins are those proteins that can be associated with either GDP or GTP. Now, in case of Rho, if it is Rho A, if it is bound to GDP, then it is in its inactive stage. But when it is bound to GTP, then it becomes active. So, if you have GTP exchange factor, so there is a Rho GTP exchange factor that displaces GDP, and instead of GDP associates GTP. So, when it associates GTP with the Rho A becomes active. Now, this active Rho A is able to actually bind to formin. Formin is an actin binding protein, and this formin is going to initiate more actin filament formation, which is required because you need to form a proper contractile ring. So, in order to form a proper contractile ring, the actin has to be formed over here. So, formin is triggered by active Rho A to form more of actin filaments to form the contractile ring. At the same time, the active Rho A is also activating a kinase, and this kinase is going to phosphorylate the light chain of the myosin. So, myosin has heavy chains and the light chain, and when it phosphorylates the light chain, the myosin gets active. Now, the myosin 2, when it gets active, it helps in contracting the actin. And so you can see how a contractile ring is actually uh, contracting and bringing the membrane, the two opposite membrane close to each other. As you can see, the furrow is increasing. So that is important. And as mentioned, the interpolar microtubules that are there are just a site for the furrow constriction. So this happens because of the Rho A, okay? And interestingly, what is also important to be noted over here is, along with the nucleus getting separated, all other organelles also that are present in the cell have to be distributed. So, for example, Golgi has to be distributed. For example, mitochondria has to be distributed. So, all of these have to be more or less equally distributed in the two cells, and that happens during the cytokinesis. So let us make the conclusions therefore. From metaphase to cytokinesis, there are levels of regulation, and that initiates the completion of the mitosis per se. So with the M-cyclin CDK aligning the chromosomes at the equatorial plane and setting the stage for anaphase, the trigger for the progress is due to the APCC activation. And in fact, interestingly, it is the MCDK that is activating the APCC. And the APCC in turn is going to inactivate whatever MCDKs are present. So once APCC gets activated, you can understand that MCDK is going to get less and less active, and they get degraded. The CDC20 binds to the phosphorylated APCC and activates it, which in turn decreases the levels of cyclins as well as degrades securin. The degradation of securin leads to pulling of the sister chromatids to the poles due to the pulling forces of the mitotic spindle, as the dynamic instability of the microtubules and the molecular motors help in the process. During telophase, the chromosomes reach the poles and the nuclear envelope begins to form. With this, microtubules, actin and myosin initiate the formation of the cleavage furrow and hence the cytokinesis. So the APCC, that is the anaphase promoting complex cyclosome, and other regulatory proteins guide the cell from metaphase through anaphase and telophase to cytokinesis, giving rise to two daughter cells, which in turn will start another cell cycle by entering the interphase. Thank you.